After you have broken down the cell walls, you are going to be working on lysing the cell membranes. Lysing is like slicing the cell membranes, causing holes in them so that you can extract the DNA from the nucleus. That's the job of your buffer. And you will be using your the buffer that you made up now, and the graduated cylinder, and your plastic bag. So follow the directions in here and then mash the fruit for 1 additional minute. The typeface did not come through too well on the PowerPoint, so please make sure that you go back to the original format of the lab in the file in this folder so that you can read up, especially with the numbers, because the numbers didn't come out too well. And go to the next one. When you are using this buffer, you are breaking up that phospholipid layer that we discussed that is between the that forms up the cell membrane by putting 2 phospholipid layers together. So when you're lysing the cells in the bag, the cellular and nuclear membranes are being disrupted. This is often called cell disruption or cell lysis by the combination of the detergent plus the pH buffer of the salt. And you can see it in here because here are your phospho groups. Here are your lipid groups in through here, and the detergent in blue can come along and break up those bonds and take them away, meaning that the DNA can come out through the holes in the cell membrane. That helps you to visualize it. Next up, we're going to be applying another physical or mechanical method to purify your sample a bit. We're going to be filtering it through cheesecloth. You'll see this on the video after this from Carolina Labs. And in doing so, if you're using a strawberry or a banana, you'll probably filter out the seeds, and you'll filter out some of the bigger materials, leaving a more purified form of what you mashed up and what you put through the detergent. That is another procedure that you would do in industry. They have very specific filters. So you're doing everything that would be done in an industrial lab. You're just doing it in your kitchen instead. But the methods have the same grounding. In fact, when we talk about industrial labs, there are basically 4 methods for homogenation. And so you are getting a cell suspension out of this that leaves most of the organelles of the cell intact if you do it right and not too aggressively. So on an industrial basis, this concept of homogenation or mixing very thoroughly so that the cell walls and cell membranes can be ruptured can sometimes involve sound. We didn't use that here. Sound waves can disrupt these membranes and open them up. We did not use that here. We did use detergents, and there are very specific buffered and detergent levels that are used. You used one of them that's frequently used. We did use filtration, much like you would do in an industrial high setting. And instead of using a plunger or a mortar and pestle, you use the bag that you molded in your hands, but you sheared the cells in the same method in order to get in a homogenized solution that hopefully kept most of your organelles intact and released most of the DNA from the nucleus. That's our goal. After you have done all of this extraction, you have to do some more purification, and you have to do some precipitation. So your purification step is going to be done with your very cold ethanol. You're going to get that out of the freezer, and you're going to get your graduated cylinder, 25 ml graduated cylinder, put about 8 mLs of the ethanol into the graduated cylinder. Then you're going to follow all of these instructions, and you're going to be adding the ethanol over about 2 mLs of your fruit solution in a test tube. Okay, so you're going to put the 2 mLs of your fruit solution in a test tube first, add the ethanol over that very gently. In the video, they don't show a gentle addition. I want you to tilt the test tube and pipette down the side of the test tube very gently so that you maintain and don't mix the ethanol with the water. If you mix the ethanol with the water, you will defeat the whole process of the experiment. So we want a level of water on the bottom and a level of ethanol above it. You'll see that in the video. All right. Very important step. And here's why it's an important step. What you're trying to do is you're trying to precipitate out the DNA. Let me explain to you a little bit about why that happens. And in fact, why it happens is also explained in your format guide from Carolina Labs, which you can refer to, but I gave you a few graphics here as well. While the DNA is in the saline solution with the detergent, the DNA remains soluble because the water is highly polar. Remember, water is the universal solvent, so the DNA remains soluble. When you add ethanol over the top of the water, the ethanol changes things. The ethanol is much less polar. So at the surface of the water and the ethanol, there's a lot going on in terms of the polarity of the two solutions. And in fact, the ethanol is less polar, meaning that the Na of the salt can get close to the DNA. When the Na of the salt gets close to the DNA, the negative phosphate groups and the positive sodium groups can bind and precipitate out. But that is right along the surface of the ethanol and the water. Your job is to let that happen. And that might take a very, very quiet area. I'm going to go get something for you, and I'll be right back to show you something. So just to back up a second on this video, because I did not realize that in the Carolina Labs video, they didn't really show you the proper way to insert the ethanol. So you're going to be using a sideways look, and the pipette is gently going to be laying the ethanol down along the side of the test tube. Right? So don't hold the pipette up and drop the ethanol in. You're not going to get good results. So your technique is to hold it on the side and gently put in those 8 mLs of ethanol. And then you can gently turn it up. If you'd like to cover it, you can, but you have a box in with your equipment that holds your test tubes. So you're going to put your test tubes into the box so that they can be held in here and nothing disturbs them for an hour or two. Then you can continue to follow the video because you're going to stick your popsicle stick in there and gently twirl the DNA up from the surface at where the water meets the ethanol. You're going to gently turn it up. Do not put the popsicle stick down into the water because then you'll mix the water with the ethanol. Ethanol is very soluble in water, and you will wreck your precipitate. So it's all about the technique. You can see it on the video pretty well. But this explains why the ethanol works, and you'll need that for part of your report sheet. And what you see here is your DNA that has been pulled up a bit by a stick, and it's been capped over there. But you'll have DNA that is lying on top of the surface between the water and the ethanol. You're going to pull it up with your applicator stick, take a photo of it, So you're going to let this sit for about an hour, and then use your stick, twirl it, take a photo, and put it into your report sheet. I hope that helps you out quite a bit. I think, oh no, I got some more for you for your report sheet. You have now made a crude extract of DNA, and you did some filtering to get out some of the bigger fragments of the cells, and you've still got a lot of other material besides DNA in your extract. You've got cell membranes in there. You've got cytoplasm in there. You've got organelles in there. You've got a lot in there. So this is what's known as a crude extract. You could further refine this extract at CCM if we were using a centrifuge. They could spin down and differentially get what you want in terms of a DNA sample. So one of your other questions on your report sheets is how you could purify this sample more. Centrifugation is the way to go. And the importance of DNA extraction, also in your format book from Carolina Labs. So you are now looking into what you can find out in terms of mechanisms for hereditary sciences and agricultural sciences, really, particularly if you're using plants, to develop more sustainable crops and crops that can utilize or utilize their hereditable characteristics as best as possible. So that's what I would say. I would follow this paragraph. You can do a little bit more research in Mastering Biology, your textbook. I have quite a few of the graphics are right directly from your textbook. Chapters 5, Chapter 6, and Chapter 11. So you can go in there if you need some more information. I hope this helps you out, and take care and have a great lab. Do good work.